Hello, welcome back. Today we're going to continue the topic of viruses. So, the virogenetics are going to be really headed or driven by spontaneous mutation. So, it can be a point mutation, frame shift mutation, whatever. And that's due to the poor fidelity of the viral polymerases. But they do this on purpose because it's kind of like it's like those fish that release a lot of eggs because even though maybe 1% of the like little fish babies survive, it's the best and the strongest out of that, thousands and thousands of eggs released. So like the viruses kind of go through the same evolutionary idea. There's a higher rate of mutation in the RNA than the DNA viruses because there is no error checking mechanism. There is somewhat of error checking mechanism for a DNA virus, but it's nowhere nearly as good as a human's error checking mechanism. So, because of that, there's a bunch of mutations that can occur. A lethal mutant is the inactivation of the essential genes. There's no replication. There's no virions replicated because these might be because of a genetic defect or the virus has infected the wrong cell. So by that I mean like one of those cells that are non-permissive, they don't replicate. So a defective mutant has a deletion or mutation of the genes, but it is able to replicate with the help of other viruses. So these would be like the adenoviruses, right? The host range mutant is going to be a change of host or tissue specificity that usually result from the nonsense mutation. Sometimes it works really well, like maybe an original virus targets muscle cells, but it's kind of hard to get into the muscle cells, right? But the one mutant that can attack like mucosa, that's much easier to get into the bloodstream, so that one would survive. An attenuated mutant is a human synthesized and altered, so we use that to make a vaccine. And conditional mutants are going to be the ones that multiply under only some conditions, such as like the perfect temperature. Recombination is an important distinguishing feature from reassortment because this is an exchange of genetic material between different but related viral strains. So there is crossing over. There is actual like recombination within a region of DNA. Reassortment, on the other hand, is when segments are exchanged. So as you can see here, this is like A, B, and then when they are like replicated, this one might have like parts of genome of A inside virus B. So we use these to make the live attenuated influenza vaccines. A genetic shift is going to cause major changes. Genetic drift is going to be only minor changes. So this generally happens over generations. Phenotype mixing is when a progeny virus acquires the coat component of the two parent viruses. So this is A, this is B, and then when they combine, you get a bit of A and you get a bit of B. Or there can be the genome of one virus surrounded by the capsids of either both the parent viruses or the envelope of one parent. So here, this would have the genome of A, but with the mixing, it can have the capsids of A and B, or it can have the capsid and the genome of B, but then have an additional envelope from B. Sorry. So it can have the genome and capsid of A, but the additional envelope of B. So there's no change in the genome. There is no recombination because this is purely like outer kind of manifestation. So this is going to change the tissue specificity. So maybe A was for muscle, B was for mucosa. So this heightens the chances of the virion entering the bloodstream and infecting. So the pseudovirion are going to be nucleic coats, and acids are going to be completely mismatched, but these are not going to be able to be passed onto the progeny, right? So you say we have the capsid and the genome from A, and then the envelope from B. So once this replicates, it's going to make A again, because the envelope from B was acquired. So that's not going to be passed down unless that was, onto a plasmid or incorporated into the genome itself, but it's not. Complementation is when a helper virus is going to help a defective virus in replicating, and it does so by making the missing components and making the proteins for both viruses. However, the packaged genome of the original defective virus is still defective. Okay, so Cincia Formation is a cell-to-cell induction or infection. So these happen when a macrophage envelops a bacteria and is unable to break it down. So, such as cytomegalovirus. They will release TNF alpha or interferon gamma to induce more macrophages to come, and then all of these bind together to make a syncytia cell. So they make a multinucleated syncytial cell, and these are going to be induced by the enveloped viruses, because the enveloped viruses are really hard to kill. Remember, because it has 2 layers. It has the envelope and the capsid. So in these cells, they're going to have something called intraplasmic inclusion bodies, which are the actual virions, or the intranuclear inclusion bodies, which are the viral proteins. Over time, though, since the body isn't able to kill these bacteria, it's going to wall itself off and form a granuloma. Okay, so the abortive infection is not ideal for the virus. The replication doesn't occur, so the virus just dies. In an inapparent infection, there's little or no damage or rapid resolution. So it's a little bit of an infection, but the host response is very strong and very fast. So there's no actual symptoms occurring. In the acute or lytic type of infection, there's going to be host cell death, and this is going to manifest with symptoms. Persistent is going to be, this is going to be chronic acute phases. So with the inflammation, the fever, the cytokines release, it's going to be over long periods of time. So it can be chronic, latent, recurring, transforming or immortalization oncogenic viruses. So in the lytic cycle, the host, the bacteriophage, the virus is going to infect a bacterial cell. It's going to make the bacterial cell replicate the viral genome and make the viral proteins. Those things get packaged, and then the cell lyses, releasing all the phage. In the lysogenic cycle, the phage will infect the cell, gets incorporated into the host genome, and then the cell divides, and then the prophage DNA is going to pass on to the daughter cell forever. And under stressful conditions, the phage DNA is excised from the bacterial chromosome, and then that enters the lytic cycle. So this can actually happen in human cells, not just bacteria. And this is going to be herpes simplex 1. So this is going to be your cold sores. Okay, so the immune response, real quick, it's going to be the activation of the natural killer cells by the production of interferon alpha and beta. Natural killer cells are going to make interferon 12 to activate the T cells. Oligoadenylate synthetase will activate N-deoxyribonuclease L to degrade the viral RNA. Serine threonine protein kinase is going to inactivate the elongation interferon 2, and MX proteins are going to block viral transcription. And interferon is going to increase the MHC class 1 expression and antigen presentation. It's going to activate T cells. T cells are going to activate B cells. B cells are going to make the memory cells, so on and so forth. In adaptive immunity, the cell-mediated helper T cell CD4 and the cytotoxic T cell CD8 are going to kill the intracellular phase of the bacteria. Intercellular phase is going to affect the presentation of the antigens, and CD4 is going to recognize it, bringing over and the B cells. B cells are going to make the antibodies, and from there, macrophages are going to come, CD8 cells are going to come, etc., etc. The humoral immunity will target the extracellular phase or the cell surface expressed by the antigen. So that's going to result in opsonization, neutralization, complement activation, and eventual elimination by the natural killer cells. The viruses will evade the immune system by creating antigenic variants thanks to mutation. It can go into latency where the host cells won't be able to find it. Infection of immunoprivileged sites, such as places that are bare from the blood-brain barrier, such as like the central nervous system. It's the spinal cord. They can also alter its antigen presentation, interfere with KL and KAR, so that's not going to allow the natural killer cells to come and attach to the defective cell or the infected cell and lyse it. And it can direct the infection of the immune cells. Okay, so light microscopy is going to observe the change in the infected cells, see if they form attachment to cultures, colonies, syncytia, see if there's any inclusion bodies in it, and whether red blood cells are going to stick to it. Hemadsorption is when the hemoglobin is expressed on the infected cell and red blood cells will stick. And in cell cultures, you want to see the cytopathic effects along with the hemadsorption. Okay, so in serology, it's a little bit more useful because it's more accurate. UV microscopy is going to identify the tagged fluorescent antibodies in order to detect and identify the viruses. Electron microscope, this is just for research purposes. Serology is going to be using the antibody to differentiate the components of the viruses along with staging the disease. So we use ELISA, rapid test, hemagglutination, inhibition assay, the neutralization test to identify viruses. We also use the PCR and in situ hybridization. PCR is going to be detecting the viruses. Let's see how fast it's replicating in the body and what is being replicated. The PCR advantage is that it's really fast and has a high sensitivity to the virus. However, it's easily contaminated and hard to set up and interpret. The ELISA is using the patient enzyme to react with a substrate to produce a colored product if it's infected, and it uses the sandwich method. So there's an antibody in the serum, and you give the serum the targeted antigen, allow those to the mix, and then you add in a tagged antibody to see if the antibody has bound to the antigen. So if bound, the substrate is going to produce a product, and it's going to make the vials cloudy, and it's going to make a color. So the darker the color, the more the product. So there is a stronger attachment of the antibodies. It can be direct or indirect, similar to the Coombs test, and it can detect the HIV P4 antigen along with the HBs AG of hepatitis B. In Western blot, you're going to detect the viral proteins by electrophoresis, and we're going to interpret bars. Okay, so viral isolation and growth, we use embryonated, so it's a fertilized egg, chicken egg in particular, along with various animals and cell cultures. The cell cultures we need to pay attention to are the primary cells, which are the monkey kidney cells, and that those can only make 1 or 2 generations. The semi-continuous cells would be the embryonic kidney cells, the skin fibroblasts, and these can be cultured up to 50 times. The continuous cell, the most important ones are the HeLa cells, are going to be your immortal cells. The application of these labs are to create a presumptive identification. We want to see how the cell is reacting to see if any like inclusion bodies have formed, hemabsorption, hemagglutination, any interference. The definitive identification is going to be the neutralization, so like whether the virus is going to be responding to the antibody or not antibody, the antivirals or not. Hemagglutination, inhibition, immunofluorescence, complement fixation, and ELISA. The plaque assay is going to be a quick and not very accurate test. It's basically the number of plaques times the dilution factors for the amount of virus in the titer, and that shows for each plaque, it's a clump of dead cells killed by the virus, so we just want to see how far the infection has progressed. And the focus assay for the virus with oncogenic potention is when the transformed cells, they grow into a little lump instead of a monolayer. So the titers are going to be a detection of early antigen fluorescent foci to detect the early proteins, and they are going to be serially diluted in order to see like either if the antibiotic is being, or I'm sorry, the antiviral is being affected or effective, or whether the immune system is working, and seeing how far the progression is of the disease. You can also use now monoclonal antibodies tagged with a fluorescent dye, really similar to the PCR method, in order for this to like identify the viral antigen. This is more for like academic reasons, really. Hemagglutination inhibition assays are going to quantitate the viruses that are expressed by the hemagglutinin on the surface. So we're going to do like basically a titer as well, and see how much agglutination occurs. So when you see like the max amount, that's kind of like how much the person has been infected, and then we dilute it with like, I don't know, antivirals or something, in order to see if like there is a minimum doses of the antivirals that can be given. So these are pretty useful in a variety of ways. The rapid test on for the HIV and coronavirus, really, it's going to depend on the immunocomplexes, so it's going to depend on the antigens and the antibodies. Okay, so that's it for now. Thank you for your attention, and good luck studying.